Hello everyone, welcome to my YouTube channel. So let's discuss about the next major topic, that is predictive methods using protein sequences. Okay, the entire model is about predictive sequence methods for protein sequences. In that this major topic, one of the major topic, under this many subtopics will come. Like first topic will be protein identity based on the composition. Under this we will be discussing about the four tools like XPASI, ACOMP-IDENT, ACOMP-SIM, prop search, mouse. Okay, all the four things we'll be discussing. Then the physical properties based on the sequences. We are having compute PI or molecular weight, then prot param, next peptide mass, T grease, SAPs. Next, under secondary structure and folding classes, we are having NN predict protein soap and P sprite. Under specialized structures or features, we are having coiled transmembrane, and then finally the tertiary structure, we are having Swiss model, DALI, and Toppitz. This is how it looks like. Around 5 different protein identification based on different things, like based on the composition, based on the properties, based on the secondary structure and folding, based on the specialized structures or features, and finally based on tertiary structures. We'll start from the beginning. Yes, introduction. Definitely the entire chapter or a module focuses on the computational techniques that allows the biological discovery based on the protein sequence itself or on their comparison to protein families. Whatever we discussed the previous topic as a major topic also comes under this. So 20 amino acids found in protein, greater diversity of structures and functions. Definitely all of you know that each amino acid residue can influence overall physical properties of a protein. Thus each residue has a certain propensities, that means the probability of influencing to form structures of different types in the context of the protein domain. Okay, so now the major precaution with respect to the predictive techniques is that regardless of this any method, okay, whatever the methods we are going to discuss now, the results are nothing but what they all are predictions. That's why it's called predictive methods. So one method may be appropriate in a particular case but totally inappropriate in another case. Understood? That's the introduction. Now the protein identity based on the composition. So as the name itself indicates, depending on the composition. Now whatever the computational tools we are using, okay, so they will identify unknown proteins based on the physical and chemical properties of the amino acids, because the composition has to be there. Now these tools are available through XPASI server at Swiss Institute of Bioinformatics, that is SIB. So two things: to assist in identification of unknown proteins isolated through two-dimensional gel electrophoresis. That is the first and primary objective. The second one is to predict basic physical properties of known protein. So how many tools are there? 4 tools. ACOMP-IDENT. COMP stands for composition, is simply AA is amino acid, definitely. Then COMP similarity. Then prop search, that is protein P search, and then mouse. ACOMP-IDENT. So rather than using the amino acid sequence to search Swiss prot, it just simply uses amino acid composition of unknown protein to identify known protein of the same composition. So there the similarity, you remember the sequence similarity, here it will be composition similarity. Exactly same words, replace sequence with the composition. Now working, how it works, so input any desired amino acid composition, like for example isoelectric point, PI, or molecular weight of the protein. These are all what composition of the amino acid, right? Appropriate taxonomy class has to be mentioned. Next, user must select one of the 6 amino acid constellations. So for each sequence in the database, whatever the sequences are there in the databases, the algorithm computes. That means it compares a score, that means based on the differences in the composition between the sequence and the query position, and finally it will give the results. What does the results will contain? A list based on all the proteins from the specified taxonomic class without taking pi or molecular weight into an account. Okay, so now the list based on all the proteins regardless of taxonomic class without taking pi or a molecular weight into an account, and then a list based on the specified taxonomic class that does take pi and molecular weight into an account. Now two things you are having, one with the molecular weight and the isoelectric point, another without molecular weight and isoelectric point. So with that difference only we can identify the comparison, or you can say that you can identify the composition of the unknown by using the known. That's how it works. Next, second one, a comp sim, that is based on the maybe the composition based on the similarity sequence identity. So variant of again a comp ident, a variant of the first version, that is a amino acid composition identification. So rather than using the experimentally derived amino acid composition as the basis of the search, the sequence simply uses Swiss prot protein. Okay, instead. So working, a theoretical pi and molecular weight are computed using compute, that is nothing but pi bar mw, that is molecular weight and isoelectric point, what the parameters we are talking about. Now that assumes that the amino acid composition across the species boundaries is well conserved. Imagine it is having a good conserved regions. Then by considering amino acid composition, the investigators or the researchers can detect weak similarities between the proteins whose sequence identity falls less than 25%. If any sequence similarity falls less than 25%, then it will be considered as a weak similarity. Okay, and that will be considering as the amino acid composition of those kind of proteins will be excluded, it will not be included. Okay, that's why it is comp sim, that means compositions based on similarity. Next, prop search. So it uses amino acid composition of a protein, again composition of the protein, to detect weak relationship between the proteins, like more robust than the previous one. Working 144 different physical properties are used in performing this analysis, not just one or two. Examples: molecular weight, average hydrophobicity, average charge, all those things. Next, collection of physical properties is called the query vector. Whatever the physical property you are collecting, like for example molecular weight, hydrophobicity, these all are physical properties. These are, if you collect these physical properties, those are our query vector. It is compared against the same type of a vector pre-computed for every sequence in the target databases by using the information already available with Swiss prot and PIR, because they act as a protein data bank. All the protein sequences will be there. So input the query sequence which you want to know the composition. The result ranked by the distance score. Okay, definitely the more the similarity, the first it will be the name. This score represents the likelihood that query sequence and the new sequences found belong to the same family. Now finally the MOS, that is molecular weight search algorithm. So it again works based on the information obtained through the mass spectrometric MS techniques. Because of the molecular weight is the concept here. So information, because composition from where you are getting mass spectrophotometry. Okay, working. So it uses both molecular weight of intact proteins and those resulting from the digestion of the same proteins with specific proteases. You take a entire protein with the entire molecular weight and then digest the same protein with specific proteases enzyme which cleaves the protein structure. So input will be molecular weight of the starting sequence and the reagent used, and resultant masses, that after decomposition resultant masses and composition of the peptides generated by the reagent. Now the calculations are based on the information contained in the OWL, that is ontology web language non-redundant protein sequence database. That means there where the calculations are what you call the the particular computer program language which involves in the creation of these calculations. So all those things will fall under input. So output will be is returned as a ranked list of the top 30 scores with OWL entry name, matching peptide sequences, and as well as other statistical information like in table how you used to get for blast. Okay, that was about mouse. So that was about based on the composition. The second thing is physical properties. Based on the physical properties based on sequence, we are having compute pi MW, XPASI again, prot param, peptide mass, T-grease, SAPS. The first one is compute pi MW. It simply works as again two parameters. So tool that calculates two parameters, that is isoelectric point and as well as molecular weight of the input sequence. It determines pi based on the pKa values. So we all know that, we have studied in PUC. Next, molecular weight are calculated by the addition of the average isotopic mass of the each amino acid in the sequence plus that of one water molecule. One water molecule has to be there. So molecular weight, you know how to determine molecular weight, you know how to determine pi. Now input is what? FASTA format, or it can be the Swiss prot accession number. User may specify a range of amino acids so that the computation is done on the fragments rather than on the entire protein. You can just go for the 1 to 100 amino acids rather than the entire sequence. Next, prot param. So based on the input sequence, prot param calculates the molecular weight, isoelectric point, overall amino acid composition, aliphatic index, other basic physicochemical parameters. How does it work on? It is not there universally, but just mentioning and their importance, their introduction, all those things you can keep. So peptides determines the cleavage products of the protein after exposure to a given protease or a chemical reagent. So here peptide itself is short piece of again protein, piece of a protein, what we call, right, determines a cleavage products. That means once the protease enzyme or maybe any chemical reagents breaks down the protein molecule, we are going to determine those products of a protein. Now the enzymes and as well as the reagents available for cleavage through the peptide mass, it can be trypsin. Okay, we are having a chymotrypsin, lyc, cyanogen bromide, arc-C, aspen, gluc-C, that is bicarbonated or phosphated. All these things are the enzymes that can actually cleave the protein to get the products of protein, that is peptide, what you can say. So input is Swiss prot identifier, result will be tabular format. It will give the two major information, that is one is theoretical pi and molecular weight of the starting protein. Next, then the mass, position, the modified masses, information on variants from the Swiss prot and the sequence of the peptide fragments. All the information will be given in the peptide mass, that is again coming from the XPASI. Next, finally the G-grease. G-grease calculates the hydrophobicity of the protein along its length. So finally we are having the different terminology here, that is hydrophobicity, that is water repellent property. Hydrophobicity, a relative propensity of the acid to bury itself in the core of protein and away from the surrounding water molecule. This all of you know. So this tendency coupled with steric and other considerations, for example, it influences how a protein ultimately folds into a final three-dimensional conformation. Depending on hydrophobicity, the protein folding structure formation will be there. So the T-grease is a part of FASTA suite of a program which is available from the University of Virginia. It relies on hydropathy scale. Amino acid with higher positive scores are more hydrophobic. If you are, for example, if more amino acids are there, if more hydrophobicity is more, then the positive score T-grease will give you the more positive score. So those with more negative score or more hydrophilic in nature? Definitely, since it calculates hydrophobicity, maximum score represents hydrophobicity, minimum score represents the hydrophilicity. So a moving average or a hydropathic index is then you can calculate with using standard formulas across the protein. Next, results plotted as hydropathy index versus residue numbers. And finally, SAPS. So SAPS, statistical analysis of protein sequence algorithm. So it provides definitely the statistical information of a given query sequence. So input definitely just a protein sequence. You download it from NCBI or SwissProt or UniProt or PI or whatever. Next, output will be a large amount of physical and chemical information on the protein. Lot of information you will get in this SAPS. So composition analysis, specifically speaking, with counts of amino acids by type. Next, charge distribution analysis, including locations of the positively or negatively charged clusters, high scoring charged and uncharged segments, information on high scoring hydrophobic and transmembrane segments, repetitive structures, multiple as well as the periodicity analysis. All the informations will be given in statistical analysis. As the name itself indicates, statistical analysis, it will definitely have a lot of data or information. That was about the physical properties based on the sequences. Now speaking about secondary structures and folding classes. The first step in the analysis of newly discovered protein or any gene product of unknown function is to perform BLAST or other similar search against the public database. BLAST, FASTA, ProSite, all those things. So if there is a statistical significant hit, definitely there may not be any information in the sequence record regarding the secondary structure of the protein. In such situations, what happens? There are methods available for predicting the ability of the sequence, how able this particular sequence is to form the alpha helix and beta strands. These methods rely on observation made from the groups of proteins whose three-dimensional structure has been experimentally determined. Definitely based on the experiments, three-dimensional data only, we can able to say that whether they have possible, whether they have ability to fold to alpha helix and beta strands. So the tools: NN predict protein, SOPMA, Cypred. NN predict feed forward neural network to assign the predicted type of the each residue. Input FASTA format is there, the folding class of the proteins like alpha, beta or both alpha, beta. Output will be residue are classified as alpha helix as H, beta strand as E, or neither will be as just a hyphen. So if no prediction, a question mark will be there. Okay, and the option line specifically it specifies n for number of folding classes of the product predictions. A for specifies as alpha, B specifies as beta, A bar P specifies alpha bar beta. Next, predict protein. So working, so predict protein sequence will be there. It will act as a query against the SwissProt to find similar sequences. Then algorithms which are called as maxome to generate a profile based multiple sequence alignment, and then it will just go with the BLAST kind of similarity search, and finally it will predict the best fit. Next, best score. Next, PSD sec, that is it assigns each residue to a secondary structure type and then provides statistics. What they represent? They represent the confidence of prediction at each position in the sequence. Why this confidence of prediction is important? Because that's how they form the three-dimensional structure. Next, input will be single letter amino acid code, that is amino acid code by NCBI maybe, so or by its SwissProt identifier, just number, accession number, or a multiple sequence alignment in FASTA format, or it can be as a PIR alignment format also. So what is the output? Reliability index of a prediction, how much reliable you are. If for example it will range from 0 to 9. Okay, so if the value is 1, it's very least reliable. If the value is 9, then the maximum confidence. Next, SOPMA, self-optimized prediction method using alignments. So it is again developed by the CNRS France, that is the protein sequence analysis server. and takes a unique approach in making secondary structure predictions rather than using a single method. It uses 5 different methods. Now the prediction from which are subsequently used to come up with consensus prediction. That means out of 5, where more common things are there or majority is there, that will be taken. Now the methods used are the we are having GOR method, that is Garnier Gibbard Grubson method, and the Levin homolog method, double prediction method, PhD method, and method of CNRS itself, that is called SOPMA. All these 5 methods will be used to predict and finally take the consensus of it. So briefly, this self-optimized prediction method builds a sub-databases of the protein sequences with known secondary structures. Each of the protein, whatever they got, is in a sub-databases, is then subjected to a secondary structure prediction based on the sequence similarity. Initially, go for the secondary, what are the different protein sequences for the known. Next, go for the structures, sequences, structures. The information from the sub databases is then used to generate a prediction on a query sequences. Now input sequence itself in a single letter format, output from each of the component predictions as well as the consensus. Each means all the 5 methods, and then finally the consensus one. Next, P Cypred, that is 5, it uses the knowledge again from the PSI BLAST searches for the input sequence to perform predictions. So again works as like similar as BLAST. It uses 2 feed forward neural network to perform analysis on the profile obtained from PSI BLAST. Now again input is same FASTA format or single letter raw format. Output will be a text file in an email message or graphical representation of a secondary structure, which we have visualized actually. Visualized using Java application called as Cypred Wave. Next, the average prediction accuracy of Cypred is in 3 structural states is 76.5%. It is actually a very good average, which is higher than any of the other methods which are described here so far. Next, we are having that was about 4 methods on secondary structure and folding classes, which is important, can be asked for separately. Next, specialized structures or features. Now, the presence of the certain specialized structures will be there in the protein, right? So those things, like for example coiled coils, transmembrane regions, that can also be predicted. There are not as many as methods for making such predictions as there are for secondary structures, primarily because the rules of the folding that induce these structures are not completely understood. That is the reason there are no much predictions tools are there to detect coiled coils and transmembrane. That's why they are called, as they are specialized one. Okay, and also they are not completely understood to identify it, okay, in different indirectly. So coiled coils, so coils algorithm is there. So query sequence against the database of a proteins which is known to have a coiled coil structures. Okay, so imagine the query sequence, you want to check it for it whether it has got a coiled coil structure or not. So probability is there, you think that there is a chance. So also it compares a query sequence to all the PDB, that is protein database subset containing global sequences, then the differences in scoring between the PDB subset and the coils database, like similarly as a BLAST what you did, it determines the probability with which the input sequence can be can form a coiled coil. Okay, with which sequence it can form a coiled coil. So input sequence data in GCG or genetic computer group, or it can be just a FASTA format. So users may select one of the 2 scoring matrices here, MTK, MTIDK. So MTK is totally based on you are having myosin, tropomyosin or keratin, whereas MTIDK, it again myosin, tropomyosin, intermediate filaments types 1 to 4, domiciliary proteins, and as well as kinesins. So coils is designed to detect solvent exposed left-handed coiled coils and then buried or right-handed coiled coils that may not be detected. Okay, so these are about coiled coils. Next we are having transmembrane regions. We are having for that again 2 things are there, PhD topology and TM Pred. So for prediction of the transmembrane regions, it relies on database of transmembrane proteins called TM-base, so which is derived from sysprot. Again, input will be the sequence in one letter code. User can specify the minimum and maximum lengths of the hydrophobic part of the transmembrane helix to be used in analysis. Output has 4 sections. A list of possible transmembrane helixes will be there. Then a table of correspondences will be there. Suggested models of transmembrane topology will be there. A graphic representation of the same results will be there. Whereas PhD topology related to the protein secondary structure prediction method, programs with PhD suite which are used in different way to make a prediction on the membrane bond rather than the soluble protein. Now accuracies that are nearly perfect. Accuracy of predicting transmembrane helix is 92%, whereas accuracy for loop it is 96%, giving an overall two-stage accuracy as 94.7%. Helix is there, loops is there. So in addition to the predicting the putative transmembrane regions, that is small proteins, it indicates the orientation of the loop regions, like how exactly they are situated with respect to the membrane. That will be seen in PhD topology. So under transmembrane regions, transmembrane and as well as coils under specialized structures. And finally we are having tertiary structures. Here under that Swiss model, DALI and Toppitz is there. So the most complex and technical demanding predictive method in the structure prediction is the structure prediction, because it is most complicated, because more data is required. The most robust of the structure prediction techniques is homology model building, or you can say threading. So out of many models, the homology modeling is the best suitable model for tertiary structure prediction. Tools: Swiss model, DALI and Toppitz. Swiss model, a program that represents automated sequence structure comparisons. So given sequence is compared with the crystallographic databases, that is XPDB. Modeling is attempted only if there is homologue in XPDB to the query sequence. I upload a query sequence, it will compare with the crystallographic database, and if at all there is a match, then only it will go for the prediction of the structural prediction. Next, template structures are selected if there is at least 25% of identity in the region more than the 20 residues along. So it has to be more than 25% similarity. So if appropriate entries are found in the crystallographic databases, atomic models are built. Because why atomic models? For tertiary structure you need to start with the atomic models. So built, and energy minimization is performed to generate the best models. Energy minimization we need to know to calculate the angle between the atom to atom, distance between atom to atom. For that we need to calculate the energy minimization. Next, the atomic coordinates for the model as well as the structural elements are returned as an email message like results. Next, DALI, that is distance alignment tool. It compares structures with the structures, looks for the similar contact patterns between the two proteins, like overlapping one structure with another structure. It performs optimization and also returns the best set alignment solutions for those proteins. So whatever you did it for the DNA sequences, here we are talking about the structural sequences. Now the DALI web interfaces will perform the analysis on two things: either two sets of the coordinates already in PDB, because overlapping has to be done, two sets of coordinates should be there, or by using a set of coordinates in PDB format submitted by the user. Next, we are having Toppitz. Toppitz expand on the PhD secondary structure method. It just expands of PhD topology. A searchable database is created by translating three-dimensional structure here of a protein in PDB into one-dimensional strings of a secondary structures. You are going to translate the entire three-dimensional structure of the protein in PDB into one-dimensional, three-dimensional to one-dimensional strings. Three-dimensional structure will be there, that will be converted into strings of secondary structure. Now then the secondary structure and the solvent's accessibility of the query sequence is determined by the PhD method. So with the results of this computation also being stored as a one-dimensional string. Now the query and target strings are then aligned After converting into one dimension, the strings of a query and target are aligned by the dynamic programming method to make the structure prediction. Now once the results are there out, the results are returned as a ranked list indicating the optimal elements of the query sequence against the target sequence, along with the probability estimate, that is Z score, what we call, of the accuracy of the prediction. So this is what we discussed so far. Total we discussed about 5 topics here, subtopics. Under that again 4 around 9 plus 13, 13 plus 3, 16, 19 different models, tools we discussed. So next topic, AI based software, small topic, that we'll discuss in the next class.